April 8th.
What does the question say he was asking?
Oh, scientists study the transcription of yeast.
No, that's not the right one.
Oh.
Let me see.
It's about RNA though.
Oh man, it kicked me out, stupid thing. Can you guys see my screen?
No.
Yeah.
Big dummy. All right, let's see.
You only have 9 kids in 8th block?
Yeah, it's a tiny class. It's
great fun.
It's my little baby class. But they can't be separated from each other on a field trip either.
Valid.
All right.
Can you blame us though?
No, you're all adorable.
It was a trip, let me tell you.
All right, I'm hiding you guys for a minute.
Mardi Gras girl.
Oh my God, that lady.
Takes pictures in the no picture taking area. Oh.
It's a hot mess.
I thought she was a chaperone, and then someone told me she worked there, and I was like, what?
Yeah, she's nutty for sure. All right,
that was so much fun.
I love him. I love Cade.
Now I can't find it. Oh, sorry, this is my first time using this, and I'm a little lost.
That's
It's okay, Mrs. Berger.
I'm lost
Do
too.
you guys know that they will not give us any credit for all this online learning that we?
What?
Yeah, we're supposed to get like credit. We have to earn credits, like doing professional development, and they won't give us any credit for this one because for learning all this stuff. I was like, what?
Why?
Oh my God, I just learned how to do all this online stuff. And they're like, no, it doesn't matter. It doesn't matter.
No.
Super lame.
Lame.
All right, so let's see. I found the assignment now. I assigned it to you. Now, how do I pull it up? This is really scary. I'm just gonna warn you guys because this thing crashes a lot, this website. So I am not exactly sure how well this whole AP online thing is gonna work. I am a little skeptical.
I think it'll be fine. Sorry.
That was a really, really scary laugh you had there.
I'm sorry, Mrs. Berger.
Maybe
I'm a psychopath or a sociopath. Okay, don't let me go then, guy, whatever.
Where are you going?
Oh, I hate
yes,
St. Louis
yes.
drivers, Mrs. Berger. They're so hateful.
All right, can everybody see my screen right now?
Yeah.
All right, so you guys all have this problem up on your computers at home?
Yeah, well, let me go out when you're recording or when you're like.
I don't even know how I did it, but somehow I did it.
Oh, well, I was in my Gmail, and then when you started, it went full screen. So
yeah,
just minimize us.
See what I can.
So, Cade, for your knowledge, this problem is scientists are studying transcription in yeast, and they created an experimental strain that produced a modified RNA polymerase containing a single amino acid substitution. Scientists determined that the maximum elongation rate during transcription with or without the modified RNA polymerase. And it gave you a couple of graphs. And then it's got 4 questions. So I'm gonna start a timer right now. And I want you guys to start.
AP 4820?
Yes.
Okay.
Has everybody got it up?
Yes, it's loading.
All right, tell me
what are the, Miss Berger, will you tell me the questions? I'm sorry.
The first question says, describe the 3 structural components of an RNA nucleotide monomer and explain the role of RNA polymerase during transcription. So you should know that. We just did that one. And then
I'm scared.
identify the dependent variables in the experiment, identify a control, justify the need for a control, describe the effects of amytin on maximum elongation. That's just going to be about reading the graph, and then state a null hypothesis.
I
So
don't remember what null is.
null hypothesis is nothing is going to change. Remember when we did that chi-square, we found out whether or not to accept or reject the null hypothesis.
Yeah.
So this is what these problems are going to be like. You're going to have some sort of graph. It's going to have some background knowledge like the RNA and the monomers and all of that, but most of it's just going to be data analysis. So does everybody have it up now except for Cade?
Yeah.
Yeah.
All right, I'm going to start the timer. I want you guys to start typing, or you can handwrite. You can either type for the real test or you can handwrite and take a picture.
Will the real test look like this?
Very similar. This is from them for you to practice. So yes.
Okay. So like
All right,
do
ready?
all 3 of them now?
Yep, all 4 questions.
Oh, 4. Okay.
Ready? I'm starting the timer. Go. Kate, you can hang up and watch later.
Sounds like a plan.
Have a good day.
Peace out, Mr.
Peace out, dude. All right, I'm going to record this for people that didn't come in. When you guys look on the right-hand side of your screen, do you see something that says question scoring and details?
No.
Hold on.
Can you see my screen?
Yeah.
How are you doing?
Is the graph still on there?
Elliot gonna need us to send
Yeah, it
flowers
is.
or anything?
Do you see where I have my cursor right now?
Yeah.
You don't have that?
Well, I just submitted mine and it's gone.
Oh, okay. Well, I'll show you guys because this one on ours has the answer, but I couldn't see
I
what
don't have
you're.
it.
All right. When you click down here, it says for part A. So this part was worth 2 points. Did all of you, when you were doing it, have a description of the RNA nucleotide? Did you have a 5-carbon sugar, a phosphate group, and a base?
Yeah.
We have to say 5 carbon.
Yeah. Do we
Um,
have the 5-carbon sugar?
I think they probably would have given you sugar. I would have if I was grading it. What about RNA polymerase? Did you say what RNA polymerase does?
That it is able to go outside of the cytoplasm and.
Well, RNA polymerase is the one that comes in and copies the DNA during
Yeah.
transcription. So did you guys, if you said something like that, you would have gotten both the points for this one. So that would have been 2 points.
Yay.
So now on part B, it says the response includes all the following. So the dependent variable, the thing that was changed as a result of whether or not you had that enzyme in there, was the elongation rate of the mRNA, right? You look back up here at the graphs. Can you still see my screen?
Mm-hmm.
Okay, because sometimes I start talking about something and you guys can't see it and I'm confused. All right, you see how maximum elongation rate, just as a reminder, I'm sure you know, but usually the dependent variable is on the y-axis, correct?
Yeah.
What's usually on the x-axis? Hello?
Yeah.
What's on the x-axis usually?
Oh,
IV.
Right, the IV, very good. Just checking your basic biological knowledge. All right, so you guys said the dependent variable was the elongation rate, right?
Yep.
So what's a control group? What'd you put?
We can't really see it anymore once you submit it.
I know, but think about it. What did you, do you remember what you said that the control
It's
group was?
the wild and experimental string.
Right, the wild type where you didn't add that at all, where you didn't have the, what's that weird word? See, this is where they'll try to freak you out, guys. So this is true on the ACT. I'm sure you saw that as well. But amanitin, whatever that is, the compound, you'd be like, well, I don't know what that is. Oh my gosh, I'm not gonna be able to answer. It doesn't matter if you know what it is. It tells you what it does. It is going to bind to the RNA polymerase and inhibit transcription. So you know that this thing you've never heard of before stops transcription, so you're not going to be able to make RNA.
I don't know if I did.
So that's the control. And then justification that the missing control is needed. Why do you need a control? What's the purpose?
Between the two.
Right, the whole, yep. The whole point of a control is to see what would have happened had you not done whatever your experiment was. So you would have gotten 3 points if you got all those 3. Oh my goodness, you guys are brilliant. Aren't you excited?
Yeah.
Kind of, good. All right, and then this part is talking about the actual, what was the question?
Right. What if we
Question.
said 6 to 1 instead of 1 to 6
What'd you say?
for the ratio?
That should be fine.
What if you said 1 to 7?
1 to 7.
I think that's wrong.
Why'd you say 1 to 7?
Well, I tried to estimate it.
Oh,
I said 2. Like the experimental strain is 2. And the wild is 12.
yeah. So that's about a 1 to 6. 1 to 7, I imagine since it's a ratio, that would give you some leeway. So remember, this was asking about the actual graphs and the actual data. So the question was the effect on the elongation rate, determine the ratio of the average maximum elongation rate for the modified RNA. So down here, if you said that the compound decreases the rate, you got a point. And if you said that it was in a 1:6, or hopefully if you said a 1:7 ratio, you'd get the point. So now you guys are up to 7 points. Oh my gosh, you're gonna get a 5. Now, I'm warning you, I did start off with kind of an easier problem for these review sessions. And then null hypothesis. I kind of gave you this answer right at the beginning because remember the null hypothesis is that the modified RNA wouldn't do anything differently. And then your justification would talk about if the active site couldn't attach to the DNA, then you couldn't have elongation. So now you would have 9 points. Does that make sense for everybody?
Yeah.
It was easy. Like,
Yeah, this one wasn't bad. I wanted to start off nicely because I didn't want you to kill me. But I also shared this with you. Can you see my screen right now?
Yeah.
Yes.
It says water at the top.
Mm-hmm.
These are where it says PowerPoint review. This is done by somebody else, which can be kind of interesting because just hearing a different voice and a different way of explaining things. I'm sure you've gotten AP emails that have told you that different people around the country are going to go over main topic areas. Have you gotten those emails?
No.
Yeah.
No?
I mean, I've gotten like the APs doing like live sessions. Or
Yeah,
whatever.
that's what it is.
Yeah.
So this is a link to that live session for water and the live session for cells. And then, because that's as far as I've gotten so far, so I'll continue updating this. I shared this with you. Some people in that AP Facebook group that I told you about have made ed puzzles. So you go through and watch a little bit, and then they'll have some questions in there for you to answer just to keep you on track. And so I put all those on there. And then this was kind of fun. You guys remember Mr. W, right?
Yeah.
He is your, can you see my, they say science music videos right now?
Yeah.
So he actually, this is kind of cool. He went through and he's got all these different reviews. And I think after this year, he's going to charge for this. But since the Corona thing, he's doing it for free. But what he does is question of the day. So each day he goes through, and I sat and listened to the one yesterday just to see what it was about. And he really does a good job of like breaking down every component of the graph or the data table so that he can really explain what is going on in the picture. And then he gives you the questions and then he goes over them. So I figure between me going over them and him going over them and then the AP Live stuff, that should be a pretty nice review for you. What do you think?
Yeah.
How often do you suggest that we study?
I would study as much as you start feeling, when you start feeling comfortable.
Yeah.
Like you're gonna see from these reviews, especially on the FRQ, not so much if you would have had the multiple choice, but especially on the FRQ, the absolute content knowledge is not a lot of the points. Look at that problem. What was it, 2 points for you knowing what the nucleotide of RNA was and what RNA polymerase does?
Yeah.
Everything else was reading graphs and understanding experimental design, right?
Yeah.
So I want you to go through and just especially do some different types, especially like day 1. That's a nasty one. It goes back to signal transduction, and right, might not feel as comfortable with that as something that we just talked about. So go through there and listen to him. I could redo it, but there's something called don't reinvent the wheel. Somebody else has already done it. We're going to use this. And he doesn't sing in these, but he does have some songs on the site if you're missing his voice. But I can do this on Wednesdays. I can do it more often if you guys, I need to know what you guys want. What would you like from me for this preparation?
I like
I'd be
it.
fine doing it every week.
Huh?
Wednesday. I like
Every
that.
Wednesday?
Yeah.
Yeah, and then, I mean, you don't have to come if you've got something else going on, and I'll record it and put it up on the Google Classroom. But that'll give you, I
How
think,
to block our faces.
yeah, I did for a lot of it, and I'm not blocking right now, but I don't care. Frustrated with this whole process. But I think one of the important things that you guys need to practice is the timing. That's why I thought this would be good today, because I know a lot of students in class have taken way longer than you would be allowed on a test. So I think that's good for you to have to practice the timing. Like I said, this was an easier one, but next time I'll do one a little harder to, you know, work your brains a little bit. Sound good?
Yeah.
All right, well, tomorrow we will talk about phylogeny. It's gonna be super exciting. I'm gonna, people were, what?
I gotta go do my master in biology.
Nice.
Oh, yeah, I
Wait,
gotta do-
tomorrow? Is there another Zoom tomorrow?
Well, Tuesday and Thursdays I was doing Zoom just for class, and then this is for the AP test. And people were saying they would rather do the notes together than just have me record it. So I was gonna try that tomorrow.
Is there a Zoom on Tuesday?
Yes.
There was?
Uh-huh.
Oh,
But
no.
really all we did on Tuesday was kind of chat because I had recorded the lecture. So I had it down for every Tuesday and Thursday from 11:30 to 12:30. I have reserved for you guys. So, and I'm going to start recording them and putting them on the classroom. So if you miss them, you can watch that way. Cool?
Cool.
Yep.
Have a great day.
You
too.
too.
Thank you. Bye-bye.
Bye.
How do I wait? Okay, there we go.